Chapter 16, question 11, asks you to classify a few descriptions as belonging to the primary structure of a protein, the secondary structure, the tertiary structure, or the quaternary structure. And remember, or recall, that the primary structure of a protein is the order of amino acids in a peptide or protein. So if you had glycine and then valine and then alanine, that would be a different primary structure than alanine and then valine and then glycine. The secondary structure talks about, once you have that chain, how it can wrinkle locally. So usually this would be in a sort of coiled pattern, like a spring, in which case it's an alpha helix, or it would be in repeating pleated sheets. Those are beta pleated sheets. So those are ways that the peptide chain can wrinkle in a local area. That's a secondary structure. The tertiary structure is how, as a whole, the protein folds, how it goes from a straight chain to a coiled up one as a whole. And the quaternary structure is once you have a couple proteins, this orange one and this blue one, how they can clump together. So here you'd have several proteins clumping together through something other than covalent bonds. So through some type of intermolecular force. So those are the 4 levels of protein structure, and we want to look at these descriptions and determine which level of structure they belong to. So A says R groups interact to form disulfide bonds or ionic bonds. Remember, R groups are the variable in amino acids. In amino acids, you always have the carboxylic acid. That's connected to a carbon that's also connected to an amine, hence amino acid. All of them also always have a hydrogen. It's the 4th thing that they're bonded to that makes the amino acids different, and that's called the R group. So if 2 R groups interact to form a disulfide bond or an ionic bond, what level of protein structure does that describe? When the R groups come together to make the whole protein fold, even distant parts fold so that the protein folds globally, that is the tertiary structure. Those R groups coming together, for example, if you had a peptide like this, and you had nonpolar amino acids on opposite ends, the whole peptide might come together so that those peptides are right next to each other, and that would make the whole protein fold, and so it would represent tertiary structure. They can do that with hydrophobic interactions like we just saw. You can do that if you have 2 cysteines, they can come together and form disulfide bonds, and if you have ions, then they can come together and form ionic bonds, which are just a type of hydrophilic interaction. Remember that an ion is any atom that has a charge. Okay, how about B? It says peptide bonds join amino acids in a polypeptide chain. So you'd have like cysteine bonded to valine, bonded to aspartic acid. When the peptide bonds, which are these bonds between the amino acids, join amino acids, they join them in a certain order. And the order of amino acids is the primary structure. So that description is describing the primary structure of a polypeptide chain. Polypeptide chain is just a fancy word for a protein. It's a chain of amino acids that has lots of peptide bonds. It says the next one, C, says several polypeptides in a beta-pleated sheet are held together by hydrogen bonds between adjacent chains. Now this one could be interpreted in a couple different ways, but because they say that there are several polypeptides, so for example, you might have one polypeptide here that's all folded up, and another polypeptide here that's all folded up, and they have beta-pleated sheets in them. They said that they're being held together by hydrogen bonds. Remember, hydrogen bonds are not actually bonds. They're 30 times weaker than a covalent bond. They're actually intermolecular forces. So intermolecular forces. And so those are two chains that are sticking together without being covalently bonded together. And that is quaternary structure. If multiple chains are sticking together without being covalently bonded, that's quaternary structure. Last but not least, D says hydrogen bonding between amino acids in the same polypeptide, and this is how this is different from part C. It says that there's still hydrogen bonding like before, but it's in the same polypeptide, and it gives a coiled shape to a protein so that it would almost look like a spring or a staircase. Well, that's an alpha helix. And an alpha helix is a local type of wrinkling in secondary structure. And so that would be the levels of structure that each of these descriptions fit into.